This video describes research at Tufts University using butterflies to explore the link between early nutrition and adult reproductive success. Researchers in the Lewis Lab reared caterpillars of the cabbage white butterfly Pieris rapi on semi-synthetic diets containing different amounts of nitrogen. Butterflies were released into large outdoor cages on the Medford campus, where researchers could measure their mating behavior and reproductive success in semi-natural conditions. In this video, we will see how these experiments address some important questions about the links between nutrition and sexual selection. It's well known that juvenile nutrition affects adult characteristics in many animals. Nutritional constraints can be particularly challenging for animals with a complex life cycle, such as amphibians, some insects, and many marine invertebrates. Some creatures are characterized by discrete larval and adult stages that often live in distinct habitats with different nutritional conditions. They have a limited time as larvae to acquire the resources necessary to shape the adult and reproduce. Like other plant eaters, the caterpillar stage of butterflies are trapped in what's been called the herbivore's dilemma. Plant tissue is relatively low in nitrogen, so butterflies must somehow reconcile a nitrogen-rich adult lifestyle with a nitrogen-poor juvenile diet. For example, Pieris caterpillars feed on different host plants with nitrogen contents that range from 1.9 to 5.9%, while adult butterflies are about 11% nitrogen. In the Lewis Lab, we investigated how low nitrogen during the juvenile stage affects larval development, adult wing ornaments, and the nuptial gifts that males transfer while mating. We found that limiting the nitrogen content of caterpillars' food caused an increase in their development times. Since larval feeding provides nutrients that must sustain a butterfly for its entire life, when nutrients are scarce, caterpillars spend more time feeding so they can acquire all the nitrogen the adults will need. There is a cost to this developmental delay, however. Adults that spend more time as larvae have shorter adult lifespans and lower mating successes. In adult Pieris butterflies, bright wing coloration is one key trait that requires a lot of nitrogen. This is because wing color is based on nitrogen-containing pigments called pterins. Females prefer to mate with the brightest males, so males need nitrogen to build colorful wings. Males also need nitrogen to manufacture spermatophores, which are large, sperm-containing packages transferred to females during mating. These nuptial gifts contain nutrients that females can use to produce eggs. Nuptial gifts also increase a male's paternity share when females remate. When we raised caterpillars on a low nitrogen diet, we found that the adult males had wings that were less colorful. Surprisingly, however, these low nitrogen males continued to produce normal nuptial gifts. Don't worry, we didn't forget about females. Females rely on nitrogen-based wing coloration to attract mates. They also need nitrogen to produce their eggs. We found that caterpillars raised on low nitrogen diets produced adult females that were less colorful, just like males. When choosing their mates, male Pieris preferred the more colorful females who, in other experiments, had proven to be the most fecund. Bearing all of this in mind, what can we conclude? Wing coloration in females is likely to have evolved as the result of this male preference. Females who are more colorful and more fecund have higher reproductive success, driving the maintenance of these costly ornaments. Because of their increased reproductive success, these females have greater access to spermatophores and therefore even greater ability to provide for their offspring. Males, on the other hand, prioritize their ability to provide nuptial gifts over wing coloration, thereby also ensuring greater reproductive success and provisioning of offspring. Because males and females' roles in mating are different, they allocate their nutrients differently, both sexes attempting to maximize their reproductive success.